Okay, so as we dive into the anatomy of the cell, this is really the lowest or smallest area that we look at when it comes to an organism. We talk a little bit about chemistry at the beginning, but for the most part, the structural aspects of the organism come down to the cells. So the structural and functional aspects of the organism are the cell, and cells are going to have different organelles or tiny organs that are going to have their own specific functions. So I'm not going to get into too much regarding evolution of cells or cell theory or anything like that. I'm going to really jump right in. But generally speaking, humans have been interested in studying cells since, well, at least and able to, with some of their tools of magnification, really hit the ground running in the mid 1800s. There was a lot of microbiology research going on, and that just carried into the late 1800s and certainly into today here, 150 years on. So, but certainly humans have had a kind of a basic understanding that there's something beyond what we see on the surface that's kind of overseeing all of this. So again, these are the basic units of all living things. Um, all functions of life are occurring because of the activities and the structures of the cells. So, your organism is what it is because of its cellular makeup. So, all of those foods and beverages you consume, all of that air and anything within it that put into your lungs or that you inhale or that your critters are inhaling and that your critters are consuming, all of that is ultimately going to be consumed by the cells of the body. And so the growth of the organism, the metabolic aspects, which are simply chemical reactions. So all of these cells are receiving chemicals. So when we look at the periodic chart or periodic table, we see a whole slew, 115 plus elements, and those elements can be put together to make compounds, and we can have all kinds of activities going on with these different elements that ultimately the cells are going to get a hold of. And so when we look at this big periodic chart full of all of these chemicals, there are really only a handful that were primarily made up of, and four of those are make up the majority of what we're comprised of. So carbon, hydrogen, and oxygen, those are the big three. And then, of course, nitrogen is another one of the biggies that is involved in our makeup. So CHO and N are the four big dogs that we're dealing with from the periodic table. Now, when we put some of these together and we get some compounds, we can start to see what are called macromolecules, which are carbohydrates, proteins, lipids. Okay, and then we start to also get into on our chart some vitamins and minerals. Okay, so those are the five major structural consumption aspects that we want to make sure that our animals and ourselves are receiving. We call it nutrients, of course, but there's more to it than just that. Those nutrients are ultimately going to be consumed by the cell. So, carbs, proteins, fats, vitamins, minerals, and then, of course, the number six one that none of us can live without, and certainly our animals can, and that's water. So we're going to be looking at water a little bit today, as well as especially carbs, proteins, and fats. So metabolism is a fancy word for chemical reactions. So anytime we consume or the animal consumes carbohydrates, if they're complex and full of, they're starchy, then we're going to be breaking those into smaller consumable aspects or sugars for carbohydrates. And glucose is the primary usable sugar for cells. So when we look at cell organelles, we're going to see one that's in charge of handling glucose or carbohydrates and metabolizing it. putting it through chemical reactions. So the digestive system is putting a lot of those consumables through metabolic changes, taking the big protein, the big steak, and breaking it down into consumable amino acids. So the cells are going to consume the amino acids from the steak or the kibble or whatever that the animal just ate. Okay, so metabolism, chemical reactions, and again, those are taking place oftentimes at the cellular level. And we're going to see where we're either building things up or breaking things down. Development, reproduction, adaptation, respiration, and homeostasis. These are all the basic functions of life. This last bullet, homeostasis, this is a primary feature of what we're doing in the clinic and what any clinician is doing when it comes to trying to determine the health of the individual. So, when the pooch comes in, we're going to take some vital signs. We want to see, especially, if pooch comes in for their checkup, we would expect to see the vital signs in the normal, healthy ranges. Just like when you go to the doctor for your physical, the expectation is that your heart rate, blood pressure, respiration rate, body temperature, they're all going to be your pupil reflexes, when they shine the light in the eye and they're looking at, there are just several vital signs that we can check at any appointment that are going to provide us an indication as to how well the critter is doing. So we're trying to determine how sound they are within the homeostatic acceptable clinical ranges. So, and all animals are slightly different. So that makes your job as a veterinary technician and potentially ultimately a vet a little more complex because you're dealing with several different types of critters. Now, from the human perspective in the healthcare industry, we're dealing with humans. And yeah, humans are different depending on age and gender and cultural backgrounds and family histories, but it's still just one organism. So your role and your job in your field is definitely more dynamic and a lot more diversified regarding the types of knowledge and differences from one critter to the next when it comes to maintaining homeostasis. However, or additionally, all of those values that we are looking at, though they're going to be different from critter to critter, the actual tests that we run are going to be pretty much the same. So again, heart rate, blood pressure, body temperature. We can do blood draws to see if there's anything going on, again, on the cellular level. We could do biopsies if we need to, if we determine that there's something potentially amiss based on the results of some of the diagnostics. So homeostasis is really what we're targeting here. And again, those ranges that are acceptable from critter to critter are going to be somewhat different. So let's see. So you'll again, part of your job, if you will, in this course as we go along is to learn the art, if you haven't already, of digging deeper and really, trying to get to the bottom of whatever it is that you're interested in. Don't just stop at whatever question is asked. Usually there are more questions after you get the answers. So, the idea is oh, homeostasis. Okay, so he's talking about, yeah, I know that. I've been to the doctor or I've been to the vet, and they always do the same thing. And I wonder what the normal blood pressure is for a dog. Well, dogs are different. Maybe a Chihuahua's blood pressure that's acceptable is different than a Great Dane's blood pressure, right? So size, breed, so even that there are going to be differences. So again, if you're curious and and then you go, oh well, how about body temperature? I wonder what the normal body temperature is for a pooch. or a cat? What kind of things are we looking for when it comes to respiration rate? When we're listening to the lungs and how much breathing are they? Because if they're 60 breaths per minute and they're supposed to be 30 breaths per minute, then that animal is probably under stress. But maybe they are supposed to be 60 breaths per minute, one per second. So again, you'll learn that not in this class per se, but you can learn it right now. And like I say, that's the beauty of having the internet at your fingertips is you can pretty much learn in real time all the time. So keep that in mind again as you go. So anyway, homeostasis is what we're looking at on the clinical setting. And really, again, the when an animal is not within normal homeostatic ranges, we say that they are uneased or diseased. Okay, so disease is when homeostasis is not acceptable, the vital signs. And we also look at symptoms, right? So signs and symptoms, other things that are tricky about your job as a veterinary technician is that, not only do you have all of these different critters and all of these different ranges with regards to homeostasis, we can check the signs, but we also can't ask them about their symptoms. So we can talk to the owner or the handler, of course, but pain is difficult to get a value on. So at a human clinic, right, the doctor says, "Oh, on a scale of 1 to 10, what's your pain level today?" With a when the Pumpkin comes into the clinic, I can't say, "Hey, Pumpkin, what's your pain scale today?" Give me 3 barks or give me however many barks for where your pain level is, right? So we can't necessarily do that. But based on vital signs, based on discussions with the owner, based on our own eyes and our own sense of what's going on, we can maybe tell that, boy, the animal appears to be, favoring his, or her right hip or whatever. Animal appears, there's like animals, they can be in pain and express it in other ways. So I've seen them with tears in their eyes, and you can tell by their eyes that they're not doing well. So, I mean, just a lot of different ways of understanding symptomatic aspects as well. But again, man, you guys are true detectives in what you're going to be doing because of all of the varying factors that you have to consider. And again, that's why, having access to, the internet and all of the wonders that it can provide does make your job a lot more enjoyable, certainly. So, diversity in appearance. Again, cells are different from one to another, depending oftentimes on their functionality. Yeah, 3 billion years ago. Yeah, they've been around a while. Prokaryote versus eukaryote. No nucleus is a prokaryotic organism, and then what we're going to be dealing with primarily are eukaryotics. These have distinct nuclei in their cells. A couple of names that you may run across, but again, I'm not going to spend too much time there. Or their cells, those of course, are microscale. So we're not going to belabor that. Let's move into some of the basic cell structures. All right. So, generally speaking, we're going to see kind of like 3 primary categories of cells and cell structures. So, cells are going to have a cell membrane. Oftentimes, we see that as a lemma. Lemma is a membrane, and then cytoplasm is going to be the kind of the matrix inside of the cell. It's primarily made of water, so we'll see that. Cytosol is the fluid, and again, it's primarily going to be comprised of water. And then we'll see cytoskeletal aspects in the form of like filaments and tubules. And then, of course, the organelles. So the structures that do a lot of the metabolizing that we see with with those macromolecules, carbs, proteins, and fats. Okay, and then the brains of the outfit, the nucleus. All right, so the cell membrane generally is going to be made of lipids, although we do see some proteins embedded within the cell membrane. So here's an illustration that we oftentimes will see. It's kind of this double fatty membrane. So these are fat tails. They're going to be hydrophobic, and then so no water going through here. We have to have channels such as this or pores where to get anything into or out of a cell. Okay, so these are protein based, call them integral membrane proteins. This one has a chain of carbohydrates. Carbohydrates are there for um, energy. So if the structure needs ATP or cellular energy, then they're going to have some carb here that they could potentially get the mitochondria to use to get some ATP going. Okay. So water soluble molecules, again, they're not going to readily pass through. So they need some sort of a channel or a pore in order to get through. Whereas when it comes to gases, they have no trouble permeating the cell membrane. Okay. So again, some of the proteins are going to be integral, so they're going to basically be embedded in that phospholipid bilayer. Some of them are peripheral proteins. So periphery just means away from the center of something. So in this situation, the center that we're focused on is the cell membrane. So peripheral proteins could be either inside or outside. Typically, we think of like the cytoskeleton, some of these filaments or tubules as being aspects of peripheral proteins. But we see some out here as well. All right. Glyco, whenever we see glyco, that just means sugar's involved or glucose. There is a sugar coating covering the surface of cells. Okay, proteins and lipids on the outer layer are attached to these sugar groups. Okay, so this is how some cells are able to join or combine together. Okay, fluid mosaic. Sometimes we have what are called cholesterol bits or rafts within that model. And so we've got a whole raft with protein involved. It's still a phospholipid bilayer all the way through. And then you also see cholesterol bits. And those are lipids as well. Phosphos for phosphate. That's what this blue circle is, a phosphate. Again, not most of this isn't extremely like pressing or important. It's just kind of again giving you a thumbnail sketch of some of the terminology that you may bump into. Okay. Caveolae, kind of like little caverns. We could find hollow spaces in the plasma membrane. So, oftentimes, again, we see that those rafts in the plasma membrane with these little a protein called caveolin. And again, IN is going to be a protein typically. So if you see a word with an IN, that's usually what that means. And so you see little caves or little pockets, hollow spaces. Yeah. So what they're used for oftentimes for like for this one, you've got those uh low density lipoproteins coming in, the LDL. They come in and they get picked up by one of these caves, and then they pinch off And so then what you have is this little vesicle that then goes over to the cell and then connects with whatever cell this might be. Some whatever organ is being fed by this blood vessel. So this could be the service of the liver, you know. Eh, Some cells are going to have flagellum or cilium or multiples. Usually we would see just one flagellum and multiple cilia. These are like finger-like projections. Flagellum or flagella, a flagellum is typically a tail. So like a sperm cell would have a flagellum. Cilia are going to be more or less for either moving particles out of an area or into an area. Yeah, cilia, oftentimes found on the surface of some sort of a tube. So cilia usually would be like the nasal cavities, the respiratory tract. They're going to be lined with cilia. And they move in wave-like motions. Flagellum. Okay, so let's go inside of the cell and look a little bit more at what's going on there. Cytoplasm, inner substance of the cell. Again, it's going to basically everything but the nucleus. So cytoplasm is going to have cytosol, which is primarily water, and then a bunch of different protein structures. Uh, we'll see centrosomes, centrioles, microfilaments, microtubules. So there's a lot of different skeletal features inside of cells. So, and what is your skeleton provides you with structure? That's really what's going on with cells too, is those skeletal features give the cell structure. They also help with mobility of a cell. All right, and then the organelles, of course. Right? Fluid of the cell, and again, dissolved electrolytes. So that's going to be those minerals a lot of times that we talk about. So electrolytes are charged particles. So we see like potassium and magnesium and zinc and calcium, amino acids. Those come from proteins. Simple sugars, of course, from carbohydrates. And then proteins that are strung together, so long chain amino acids, those are going to be primarily enzymes. Yeah, those are primarily enzymes. In fact, when we look at chemicals of the body of the critter, they come in really three main categories. The chemicals are either going to be enzymes, which are protein based. They usually end in the letters A, S, E. Okay, so enzymes. Enzymes speed up chemical reactions, so they're very necessary in the cell to get things to happen at a rapid pace. So enzymes are a chemical. And then hormones, we'll do a whole chapter on the endocrine system. That's another group of chemicals. And then neurotransmitters. And neurotransmitters are going to be the nerve chemicals. So they basically are going to encourage something to happen or discourage something from continuing to happen. So it's basically the nervous system is works in kind of like a shut on off switch. So we're going to see neurotransmitters performing a lot of that action. So those are the three main chemical categories: neurotransmitters, hormones, and enzymes. Okay. And enzymes are protein based. Hormones actually are primarily protein based. Usually we think of hormones as being lipid based, but I think there are, I don't know, 5 to 10 seven, maybe 5 to 7 hormones out of like 60 plus hormones that are actually lipid based. The rest are primarily nitrogenous based. So they're going to be like protein based. So, and by the way, when we look at carbs, fats, and proteins, carbs and fats have the same three ingredients: carbon, hydrogen, and oxygen. Whereas proteins have carbon, hydrogen, oxygen, and nitrogen. Yeah. The way I remember that is protein ends with an N, so it has nitrogen with it. Okay. Cytoskeleton, again, that's that framework, 3D framework. It's going to provide some structure and support to the cell, and it also helps with locomotion. There's like an external connection too, that can enable the cell to connect with other cells. And microfilaments, we see these in skeletal muscle. We have intermediate filaments as well, and then microtubules. Okay, so the skeleton of the cell. All right. Another type is the centrosome. We definitely are going to see centrosomes and their centrioles and asters. These are going to have to do with cell division. So when the cell's ready to go through mitosis, we get the centrioles involved. They duplicate themselves in preparation for cell division. So typically a cell won't have multiples in. It might have one batch of centrioles, but if we're using an electron microscope, we see multiples, then it's probably an indicator that that cell's getting prepared to go through cell division. And again, it depends on the cell because some cells, depending on the type, have a lot. Microvilli, cilia, flagella. So cells that have these as what's their primary features on their cell membrane, you might find more centrioles in that cell. All right, anyway, organelles are little organs. So we'll move on from cytosol and move into organelles. Little organs, so these are the main ones that we see, especially these first handful. These first three are going to deal a lot with those carbs, proteins, and fats. So mitochondria deal with the carbs, ribosomes deal with the proteins, and ER deals with proteins and fats. If it's rough ER, it deals with proteins. Smooth ER deals with lipids. Okay, rough ER has ribosomes. That's what makes it rough. And that's going to deal with protein. Basically, it strings together a bunch of amino acids. So that's what the rough ER is doing. Okay. Then we'll look at these other ones shortly. Golgi is a packaging facility. All right, so real quick, I'm going to zoom through these organelles. Mitochondria are going to be, again, the powerhouse of the cell. These are going to take glucose and oxygen and get ATP. So ideally, when I add glucose and oxygen to one of these, I'm going to get 36 molecules of ATP. And ATP, again, is cellular energy. So if a cell has a pump or a vesicle, it's probably going to need energy. Hmm. So mitochondria, I know there's a lot of other info about mitochondria, but I think again, for like your purposes, just knowing mitochondria deal with carbohydrates, sugars, and help with cellular energy. On a clinical level, if an animal is deficient in energy, they're sluggish, we have to look at their consumption. Are they getting enough carbohydrate? Are they getting the right kinds of carbohydrate? So it's about amount and type. Hey, the other thing you need to make sure of is that the animal's breathing properly. So that's why we look at respiration rate, because again, oxygen is crucial to getting ATP. Hmm. I just need to take a deep breath, get some oxygen. Yeah, Brandon.
Can an animal be like deficient in something but have too much of something like simultaneously?
Yeah, absolutely. In fact, that's oftentimes typically what we'll see. So when we're looking at like homeostatic ranges, when the animal's healthy, they're generally going to be kind of like in the middle, or and then of those ranges, like there's like like areas in those ranges where that are acceptable. But if they're starting to get toward the edge of unacceptable ranges of homeostasis, and maybe even out of the range, at that point, we're going to determine, is this a hyper or hypo issue, or at least try to. So does, in other words, we might have too much of a certain chemical and not enough of another chemical. So there is this balance that takes place that certainly we could be deficient in something and excess in something else. And oftentimes they're part and parcel. So, so if we look at like at diabetes or blood glucose or something like that, the insulin levels that we look at are going to be in conjunction with what we call the glucagon levels. So in a critter, when I have excess glucose in the bloodstream, I usually will find that they don't have enough insulin. So they're deficient in the insulin, but they also may be excess in something called glucagon, which kind of like triggers the need for more insulin when there's excess sugar in the blood. So it's trying to clinically, we try to figure out kind of like which one's kind of controlling the other. So, if I can tell the glucagon to stop doing what it's doing, for instance, because again, glucagon gets released when I don't have enough blood sugar. And so the liver will start releasing a bunch of glucose into the bloodstream. So what happens after that is I need insulin to pull all that glucose out of the bloodstream. So, what may happen is if I don't have enough insulin to accommodate all the sugar in the blood, then I have potential for diabetes issues. And long term, that could be problematic for other organs. So, we need to determine maybe something's going on with glucagon. They both come from the pancreas. So, we could have a problem with the pancreas where the glucagon is. Where the body is constantly perceiving that it's low in blood sugar. So then glucagon gets released constantly, which then triggers insulin to get released. But I just don't have enough insulin because there's such an excess amount of the other one. So, or we could potentially see that there's the perfect amount of glucagon and the perfect amount of glucose. Unfortunately, the pancreas cells that produce insulin, they're called islet cells, they've maybe gone dormant or they're overworked or they're just not strong enough or whatever to pump out even just a normal amount of insulin. So yeah, absolutely. So that's really kind of the name of the kind of the name of the game in the clinical setting when we're looking at homeostasis and when the critter's out of homeostasis. And again, like what I just mentioned, glucose homeostasis is probably one of the most important types of homeostasis that we're assessing in the clinic, especially if the animal is is expressing like very sluggish, lethargic behavior when that's not what they normally do. Um, the other thing we look at heavily is calcium homeostasis. Because again, if and that relates to the thyroid, believe it or not. So, there's a thyroid and has four mini thyroids called parathyroids kind of in the posterior behind it. So if I took the thyroid out and look behind it, there'd be four little kind of like mini M&M size glands called parathyroid. And so these two glands, the thyroid, parathyroids, they work together as antagonists, if you know if that makes sense. In the same for calcium homeostasis. So for in the blood, if I don't have enough calcium, then parathyroid glands get triggered to release parathyroid hormone. And that's going to go to the bone and say, hey, we need calcium from the bone. And so that'll trigger some osteoclasts to start chiseling bone. We release calcium in the blood, and now I've got high blood calcium. So then I need to bring in a thyroid hormone, this time called calcitonin, that's going to tone out that calcium or pull the calcium out of the bloodstream and put it usually into the muscles. So calcium is kind of stored in bone, but it's used by muscle, if that makes sense. So bones aren't really using calcium, they're storing it, whereas your heart, your intestines, your skeletal muscles, all of those are um, in dire need of calcium all the time. So um, so again, blood calcium and blood glucose homeostasis, those two areas are extremely crucial to the vitality of the individual, the critter. So, and yes, absolutely these values clinically that we're looking at and assessing, whether it's, from a complete blood count, whether we're able to dig deeper and do some sort of, hormone assessments or other, chemical deep dives of the body, we would likely see this kind of teeter-totter effect where, you've got excess over here and deficient over here, and we're trying to kind of balance everything kind of in this range. So I think that's, that you're absolutely right. And picking that up from all in amongst all of this jibber jabber that I've been doing, that's excellent on your part and everybody else's too, because that's really you're getting it when you see it in that way. It's kind of like yin and yang, or on and off, or up and down. There's just this kind of, we call it hyper hypo. So hyper is excess amount of hormone or whatever we're talking about, and hypo is deficient. And so when one's out of whack, one hormone or enzyme or neurotransmitter, usually the antagonist is in excess. So, and then to try to figure out how to get one, like which one's triggering the other one to be in excess or to be deficient, or maybe it's neither. Maybe there's an external factor somewhere else that's just something in the neurological setting that is instead of this, because I we equate like hormone control and regulation to like having a dimmer switch versus a light switch. A light switch is on or it's off. And so with hormones, we like it to be more of like a dimmer switch. So you have like a little bit of a spritz. Okay, that's enough. Let's back it down because we still need a little bit of light slash spritz, but just not that much. And then, oh, now we need a little more because we're in this situation. So we're going to crank up the dial a little bit. Do, 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 do. And meanwhile, the other one over here, we're cranking down that dial. You see what I mean? There's like, it's not flipping on, off, on, off, on, off. It's more like dialing, keeping your switches dialed properly, your dimmer switches or so anyhow, yeah, so that's what you're going to be kind of looking at in the clinical setting and having that mindset for sure, because a lot of what we do is very much like critical thinking and problem solving and doing these kind of like you know um, just exactly what you're saying, putting these pieces together. It's like puzzle after puzzle. So yeah, proteins. Again, ribosomes are going to be working with proteins, and proteins, again, make up, depending on the animal, can make up a very large portion of their caloric intake. So, so anyway, ribosomes will be very busy. They're going to be basically when we eat protein, we break it into amino acids. And then we send those amino acids to the cells. And then the ribosomes work with the RNA, the messenger RNA, to kind of decipher, the recipe that needs to be strung together. And then the rough endoplasmic reticulum with these ribosomes that are attached or bound, they're basically membrane bound ribosomes on the endoplasmic reticulum, make it rough ER. That's kind of the assembly facility for amino acids becoming proteins. And a protein is either going to be um, it could be a chemical. So protein, we add amino acids into a cell, and the rough ER might be converting. It's based on the directives from the nucleus. So the nucleus takes a photocopy of the DNA, and then that photocopy is kind of trimmed by the messenger RNA. We call it transcribing in the nucleus, and then the messenger delivers the photocopied recipe chunk, to the factory, the blueprint, if you will. But again, it's a photocopy. And then out in the cytoplasm, the ribosomes, along with that messenger RNA, they're going to start trying to translate that message. So we have like transfer RNA that's going to help to translate, and so then the messenger RNA is no longer needed. And then at that point, we start to kind of replicate whatever that recipe called for. And so it could be, an enzyme, because a lot of, like I said a minute ago, a lot of the proteins that are directed by the nucleus to fulfill, the job order, if you will. So the nucleus gives out the job orders, basically. So if the job order that the nucleus gives is for a specific enzyme, then the body is going to have to gather the proper amino acids to string together to make that enzyme. A lot of our hormones, like I said earlier, too, are protein based or amino acid based. So maybe the body needs, more of a certain hormone during a certain time period, and so the nucleus is going to have the recipe for that hormone, and so it's going to make a photocopy of the recipe, and then we're going to get these RNA delivery systems, and then the ribosomes and the rough ER, are going to put these, basically they're like beads, or put these strands together and end up with some sort of whatever the hormone was that was requested by the nervous system. The other thing that we could do with proteins is string them together and get structures, like structural proteins instead of chemical protein. So, like your skin and your hair and really all of most of your organs are structural proteins. So, like collagen and keratin and those are the off the top of my head, the big dogs. But there's that you have probably heard of too. So those are protein derivatives and their structures. So and again, the cells of the skin are reliant on getting the right types of amino acids so that they can repair, the string together those amino acids to get more collagen or more keratin. So, so anyway, that's what's going on with proteins and ribosomes. Cool. Yeah, looks like we lost a cut. Yeah, it's already been an hour. Hard to believe. So I'm gonna keep her moving. Get you guys out of here a little early. The because the main things the again the carbs, proteins, and fats. Like what organelles deal with those? Because that's what the animal's consuming. They're reliant on the cells are reliant on those. The carbohydrates are going to be for the mitochondria to convert that to energy. So oxygen and glucose, equal energy, which we call ATP. So that's what happens with the carbohydrates. And then the proteins I just got done talking about, going through. And we can see then rough ER is it's rough because of the presence of ribosomes. So there's a lot of R's in this. Protein itself has an R, but you've got rough ER, ribosomes, RNA. So they're all dealing with proteins. This is protein synthesis, production. This is the production facility. Okay, it's basically a factory. And, when it comes to factories, I'm going to bring up the packaging center because that's a different area. This is simply the factory. They assemble here, but they don't package here. Okay. And then smooth ER, which is this one, notice this is rough because it has ribosomes on it. So rough equals ribosomes. Okay. Smooth is no ribosomes. So these are going to deal with fat synthesis or lipid synthesis. Uh, in muscle cells, we store calcium in this smooth ER. Okay. So anyway, these are both factories for assembly. Assemble lipids from fatty acids, assemble proteins from amino acids. Okay. So then what we end up with is this specialized packaging center called the Golgi apparatus. Ooh, Golgi packages. So yeah, if we got any supply chain people, you could start a business, maybe a supply chain for packaging medical supplies. Could be called Golgi Packaging Inc. But anyway, that's how I remember Golgi has a soft G and package also does. So Golgi packages. And again, it's going to be packaging material that's been produced by the rough or smooth ERs. So again, proteins or fats are going to be packaged. But we also have another, I guess you'd say, couple of, delivery doors to the Golgi apparatus, and that's going to be the garbage or the waste doors. So Golgi also receives and packages waste for and recyclables for delivery outside of the cell. Okay, so the Golgi is packaging groceries and garbage and product. You see these vesicles. Vesicle is one letter off from being a vehicle. If we take the S off a vesicle and put an H in, we have the word vehicle. Vesicles are just that. They're vehicles that transport the goods to and from the Golgi hub. So this could be, the Amazon warehouse shipping and receiving. And then these are the factories where the product is made. All right. And in between, even down here, from rough ER, driving that protein over to the Golgi so it can get packaged and then sent out of the cell is extremely important. So Golgi probably has some quality control aspects too, I would imagine. Otherwise, why wouldn't the vesicle just pick up the completed product and just take it to the membrane and drop it off? Why do we need this Golgi middleman? Well, there's definitely a reason for it. All right. Anyway, Golgi, package and distribution. All right, lysosomes. Lysosomes generally are going to be destroying things that aren't supposed to be inside of a cell. Although some lysosomes will break down food stuff that, macromolecules, carbs, proteins, fats, that made it into the cell. Typically, by the time that stuff gets to the cell, the enzymes have broken it down as far as it'll go. Yeah, lysosomes. They'll connect with other vesicles, kind of hook up with another truck, and then dump the load off and pick up some peroxide or something to kill whatever it is, break these big bits into smaller bits. So what's showing is this cell, so food came in, food's still in here, it got picked up by a truck, and we drove the truck heading wherever, and say this is, this could be a strand of starch. So or maybe this is a couple of you know, just I don't know, a couple bits of protein. So we just add the lysosome might add an enzyme in there, and it's going to break those into digestible bits. Peroxisomes, I guess when you see peroxide, you think of like maybe kind of a cleaner, antibacterial type of chemical. So that's really what we have here. Detoxification, removal of free radicals. That's what peroxisomes do. I don't need to know anything about that or that. All right, we're just about there. The nucleus is again, that's the DNA. It's the brains of the cell, the control center. It is the largest organelle inside of the cell. Eh, Most of our cells are going to be multinucleated. Eh, So a lot of cells. Now, red blood cells are unique. They don't have a nucleus, so there's no genetic material per se. Blood cells are formed in blood, or blood. Blood cells are formed in bone. So bone marrow is where blood cells are formed. So that's where the nucleus and they don't go through cell division. So usually a cell that needs to go through cell division would require a nucleus. Now, other cells don't go through cell division, but they have a nucleus, right? Like a neuron doesn't divide, but it does have a nucleus. But so that's not the determinant. But anyway, red blood cells live about 120 days. And so once they get a little bit overworked and worn out, kind of like, tattered, oh God, I'm done. I've just carried oxygen for 120 days. Ugh, I'm tired, right? So the red blood cell will end up well, they always are passing through the liver, but they will pass through the liver one last time and not make it out the other side. So they'll enter the liver, but not make it out of the liver. And so, if they have a damage to the cell wall, any misshapen red blood cells will get picked up as being, you know, um, unnecessary or unusable, if you will. So, so they don't go through cell division. They're just going to get lysed. So the liver will destroy red blood cells that your body no longer uses or needs. Um, and you're left with some byproducts. The body likes to recycle actually. So when red blood cells are destroyed, your body's left with some pieces that are used for other things. So one of those is bile. You've probably heard of bile. It's produced by the liver, stored in the gallbladder. And bile has kind of a yellowish orange color to it. And it gets that yellowish orange cover because of the destruction of red blood cells that takes place in the liver. So they're left with what's called bilirubin. And bilirubin um, is in essence, it's like a color, a pigment that's kind of orangish yellow that gives bile its color. So a lot of times, chemistry relies on like color, like chromatography, in order to accomplish certain functions. So the color of bile, it comes from the destruction of red blood cells. So that pigment comes from bilirubin, uh, which is from destroyed hemoglobin in a red blood cell. So Jaundice can occur when the skin turns orangish yellow, the eyes, the whites of the eyes get orangish yellow, and that's could be because the liver's destroying red blood cells at too rapid of a pace, so the body's left with a bunch of yellow pigment. So that's something we look at with our animals and with our patients. We look at the whites of the eyes, and if they're yellowish orange color, um, not always, but it could be an indicator of jaundice, which is again. So then you want to ask, oh, well, why would the liver be destroying red blood cells at such a rapid pace? Is it because there's too many red blood cells? Possibly. Maybe there's so, the liver's used to receiving, I'm just use hypothetical numbers. The liver's used to receiving, 100 red blood cells a day, and it just, or used to destroying 100 red blood cells a day and getting 100 red blood cells worth of bilirubin a day. So 100 red blood cells worth of yellowish orange pigment every day. And from that, it produces bile and it's got that homeostasis down. Well, now all of a sudden, the liver's receiving, excess numbers of cells. They all look good, but there's just too many of them. So now it's going to do 200 a day instead of 300 a day, or instead of 100, say to 200 instead of 100. So now it's doing double the amount of lysing of red blood cells. So you're going to call it hemolysis. So if the liver is doing double the amount of hemolysis, you're going to have double the amount of bilirubin and double the amount of pigment. So why did the liver do 200 today instead of 100? Well, because it got hit with 10 million total today instead of 5 million total today. So it's possible that the liver just received an excessive amount of blood. And so, in order to balance out the numbers, it was destroying healthy red blood cells. Just they're just too many, so we had to destroy them just to maintain our numbers. So that would and remember where our so then why would I have so many red blood cells? They're not even damaged. They're perfectly healthy. Well, red blood cells, remember, are born in the bone marrow. So shoot, now the bone marrow is producing an excess amount of red blood cells. I might have bone marrow cancer at that point. Well, not me, but I might have a bone marrow cancer case on my hands. So, um, but maybe the liver is doing, maybe it's excess, maybe the red blood cell count is normal. So we do a CBC on the animal, and the CBC is normal. There's the proper amount of red blood cells, but the liver, for some reason, is deciding to have recognition issues. So it's recognizing healthy, maybe red blood cells as being old, aged, or tattered. So it destroys healthy red blood cells, thus diminishing the count overall of what would normally be there. So again, so it normally does 100 a day out of 5 million. And today it's going to do 200 out of that same 5 million. So now 100 extra healthy red blood cells are gone. And so, yeah, that's going to trigger then, that's that hypo. That's going to trigger then the bone marrow to say, hey, we need more red blood cells to be pumped out because we're lacking. So, anyway, my point is uh, that, yeah, the red blood cells, um, and they don't have a nucleus. They're produced in the bone marrow, and generally speaking, it doesn't mean that it can't be reproduced. Usually, when cells have a nucleus, they don't reproduce. Red blood cells don't have a nucleus, but they do reproduce. And red blood cells also have heme in it, which becomes iron, which can also indicate whether the critter has low iron. So if a critter has low iron, it could be an indicator they have low red blood cell volume. So And then we have to look at the liver. Is the liver destroying red blood cells? And then we have to look at the bone. And maybe the bone isn't producing enough red blood cells. Why do I not have enough red blood cell? So anyway, as you could probably figure, red blood cells, their numbers, their behavior, their function, is all extremely important on the clinical level. All right. Anyway, nucleus, anatomy of the nucleus. It has its own membrane. It's almost like a cell in a cell. Even has its own cytoplasm called nucleoplasm. And then it has some chromatin, which is going to have to do with the DNA structures. And then the nucleolus, which is a tiny nucleus inside the nucleus. Okay, so again, that nuclear envelope is also a lipid bilayer. DNA and the genetic material of the nucleus, foundation for the storage of genetic information. So we're going to see all these nitrogenous base pairs, A's and T's, going with C's and G's. And so you see A's and T's go together, and C's and G's go together. And then in RNA, we don't have the T going with the A. We have a U. Uracil goes with the A. So if you're, and these are abbreviated A, C, T, G, and U. Yeah. So if you ever see a strand of letters put together and there's a U anywhere in that string of letters, that's got to be RNA. If there is no, if there's a T in that string of letters, it's DNA. So those are the two things you'd be looking for to determine if it's DNA or RNA. Yes, adenine, thymine, guanine, and cytosine. And again, you're not going to have the T, you're going to have a uracil, a U with RNA. And the chromatin, that's the genetic material. And the chromatin strands, these are what divide during anaphase of mitosis. So during metaphase, we need to line these chromosomes up, and then we need to split them. We need to divide them. So this one needs to go that way, this one needs to go that way. And so then we're going to have two daughter cells with identical uh, DNA. And nucleoli, one or more small dark staining spherical patches in the nucleus. This is where the ribosome subunits are made. The ribosomal RNA is made. All right. So that's your cell stuff. I'm going to get into cell physiology. I wanted to, oh, good. You've Rich. I'm just going to spend a couple minutes on cell physiology. Normal hydration, again, carbs, proteins, fats, vitamins, minerals, and water are the big six. And again, consuming normal hydration, consuming the same amount of water that's lost. So, believe it or not, if you drink, so I've got probably a 12-ounce glass of water. I'm almost done with it. I get time to start to think about finishing it. So that 12 ounces of water I've drank in the last hour or two, I'm going to be peeing out that same, well, not that same, but that same amount of water. That's how I keep my fluid balance, right? So animals take in water, moist foods, drinking fluids. Okay, animals lose water, so metabolic water, water of oxidation. Okay, so animals, we're all getting our water from our foods and beverages. How do we lose water? Insensible water loss, um, diffused passively, so peeing, diffused passively away from the skin, so kind of sweating or vocalizing, urinating, defeating. I think this is supposed to say defecating. I think there's supposed to be a C right there. Huh. Defeating. Yeah, pooping, defecating. Excessive would be vomiting, diarrhea, excessive sweating, bleeding, because your blood is about 55% or so water, and then elevated body temperatures. When we look at body fluids, we have intracellular fluid. That's going to be fluid inside of a cell. So if this is a cell, this is intracellular fluid. And then outside of the cell is what they call extracellular fluid, which has a couple different names. We oftentimes see it called interstitial fluid. All right. So you can see we've got a blood vessel, a capillary delivering nutrients. So dropping off oxygen into this interstitial space, which then travels into the cell itself, into the intracellular fluid. They abbreviate this ICF and ECF. Solutes are the particles. They could be solids, liquids, or gases, but it's the particles that are going to be contained inside of the solvent, which is the liquid. The solvent is going to be oftentimes water-based, and so the solutes would be, like salt inside of water. So to have salt is the solute, water is a solvent. Together they make a solution. Okay, and electrolyte concentrations normally inside of the cell, you're going to have a lot of potassium, and outside the cell, a lot of sodium. So these two are antagonists, if you will, or opposites. You're going to find sodium out, potassium in. Now, it's not going to be 100 out, 0 in. You're going to have a few sodiums, of course, is leaching in, a few potassiums, leaching out. Well, you know, so. Particles are going to move toward an area where there are fewer particles of the same. So sodium goes where there's less sodium. Potassium goes where there's less potassium. So sodium lives outside the cell. So where's there less sodium? Inside the cell. So naturally, some sodiums is going to be leaching into the cell. But I don't really need it in there. So let's say normally there are 5 in here and 95 out here. That's my equilibrium. Well, what if an extra 1 gets in here? Now I've got 94 here and 6 in here. Well, we don't like that. That's not equilibrium. So this 6, we'll say uh, 6, we'll just use that. So let's say that then this guy needs to get back out here, but he won't move, or that molecule won't move out here because this is a high concentration of sodium. So that piece of sodium wants to kind of stay in here, even though it's not really wanted in there. So it has to get pumped out. So that's what we call a sodium potassium pump that's going to work to move particles against sodium against its concentration gradient. So it's going to move sodium out of the cell. And actually, it moves 3 at a time out. So there's a 3 sodium per pump gets pumped out. Now, potassium's kind of the same way, just flipped. So normally there's a lot of potassium inside the cell, but what happens when you eat potassium? Well, now there's a lot outside the cell. Well, how does that potassium get in here? If there's more in here, we got to pump it. So they've got this kind of co-transporter. We can pump 3 sodium out of the cell and 2 potassium into the cell, okay, with every sodium potassium pump. Okay, pH. Again, the H in pH deals with hydrogen ion concentration. So the lower the number on the pH scale, the more hydrogen it has. It's like a negative base 10 formula. So, which means we're dealing with decimals. So, like if I have 10 to the negative 1 power, um, that's zero point 0.1 written out as a decimal. So 10 to the negative 2 power is zero point 0.01 written out as a decimal. 10 to the negative 3 power is 0.001 written as a decimal, and so on. So when something has a pH of 1, it means it has a hydrogen ion concentration of zero point 0.1 mole. Okay? That's a lot of hydrogen. So we'd say it's acidic. So once we get to 7, we're at what they call the neutral amount of hydrogen ions. That's water. And then above that is alkaline. So you're going to have fewer hydrogen ions and more free OH negative ions. So pH concentration and electrolyte concentration and pH go hand in hand. So again, if I don't have enough sodium or if I have too much sodium, it can alter pH. Yeah. So it's again that hyper-hypo thing. Try to determine if they have enough or not enough. If they don't have enough or if they have too much. Am I deficient or excess? All right, we're gonna do this next week in class. I'm gonna talk about diffusion and osmosis. That's labs 3A and 3B. So that's what this gets into. So we'll stop there. All right, so that's really the main thing I wanted to get into today. I wanted to discuss cells